This lesson is on the mitochondria electron transport chain. You can clearly see that I've built the inner membrane here to start the lesson. There's no need for me to have the outer membrane for this particular lesson just because I need the space. And you can tell from your legend I've set up the 6 proteins that are involved in the electron transport chain. And I would strongly recommend that you have your students kind of make like a chart where they have the names of these proteins and space for before, during, and after diagrams, and actually another column that we would call words. This is kind of a classical lesson to teach the students those types of combinations of pictures and words. So we're going to begin the lesson with the coenzyme NADH, and we all know that was produced during the Krebs cycle. So I'm going to show you a couple of moves here. Notice where I'm placing my hands. The kids can see that the N, so they know that that's NADH. And as we move NADH up to NADH dehydrogenase, we're going to conduct that redox reaction. Now, we know that when hydrogen is liberated, right, it creates a proton. So if I back this up, what I did was, these, as you know from the beginning of the kit, the electrons have the power magnet on them. You can then take the proton, that'll clearly stick to the electron, and place the H over it. So it's kind of like we have this flip move that we use. This is like a flip move. So when you conduct the redox reaction, you take both of them off and then just take the hydrogen off, and now we have our proton. And you can take the electrons and place them into the protein. Now that's one technique using the coenzyme. Now these power magnets, if you've played with them, they are strong. So to put those two electrons on NAD here, I mean, you can move it, but one thing that I like to do is very simple. You take a transparency that you all have to either photo. This is a transparency that we photocopy on. It's a little bit thicker than the ones we write on. Just take 4 fairly small magnets that you can see here, and you place the transparency. Now becomes an interesting way of moving material with the model. And I can show the students, so there's NAD. So now I can start to move NAD very fluidly. I can place the electrons at different points so they can actually see all of the language. Place the proton back on, and I can place the hydrogen back on. So this particular technique is something that I developed by accident and I really like a lot. So it's simple for you to do, and give it a try. So you can see how much easier it is for me to move that. And we just conduct the same reaction. Now we're going to pump that proton. So there's one proton. Q, you do a little dance move here. Is going to shuttle those. And again here, I got to cheat a little bit because if this protein is in the membrane, I'm not going to be able to slide it. So I kind of shuffle it near here. It delivers the electrons to the cytochrome complex. We're going to pump another proton up. C does the dance, picks up the electrons, shuffles the electrons over to the oxidase complex. One of the things I really like about this particular model, again, is showing the students here, and the reverse is true in photosynthesis, is the connection between oxygen and water. And it really blows them away when they see, when we finish this reaction. Because as you all know, when you look in the textbook and you see 1/2 an oxygen, the way it's described most of the time, it's actually written in that fraction, 1/2 of an oxygen. And the students are very confused by that kind of notation. So to model this, the oxidase complex is going to pump another proton. I move O2 into the active site, and split O2 into O. Now, I'm just going to leave that for a minute because you need some protons here. So we have 1, and you can talk about the concentration gradients and everything. 1, 2, 3 protons that we're going to get a return on those 3 protons, as we all know from that one redox reaction. We're going to synthesize 3 ATP. So we now have some protons in the matrix. We've synthesized some ATP. So when the oxygen attracts those 2 electrons, 1, 2, it's going to pick up those 2 protons now, 1, 2, and then that's when you can do the flip move and show the kids, voila, that oxygen molecule is now a water molecule. And that kind of brings that whole, obviously, end of those electrons into a water molecule story. Now, in terms of the manipulations, you really have everything you need to know about the rest of the story, right? You're going to conduct another redox reaction with FHD2, right? And you're going to have another NAD outside the mitochondria, an NADH from glycolysis. You could build, you've got 1 extra protein here. So this could be your shuttle, right? So you could bring that NADH in. You could shuttle those electrons onto FAD and continue the reactions. However you want to frame it, you understand how I do my manipulations for the electrons, for the redox reactions. You get a chalk ATP and you're producing water molecules. And it's a really fun lesson to teach because it's really crystal clear and the kids get very focused on the before, during, and after diagrams. And they certainly have a lot of questions here. Sir, what about this oxygen? And again, I mean, as you all know, that can lead right into free radicals, right? And you can actually start now talking about some genetics and all kinds of fun. So this is one of my favorite lessons to teach, and all the best. So that gives a conclusion to the electron transport chain mitochondria lesson.